Recording in progress.
Okay, good morning.
Good morning, teacher.
Good morning, teacher.
Good morning, all of you. Hi. So last time we started to discuss uh, about the chemical substances, right? I will open from here. Hi. So, what can you remember? We mentioned about the main one other major biological molecules. We mentioned about carbohydrates, and we classified carbohydrates, right? We classified carbohydrates into three categories. One are the monosaccharides, disaccharides, and polysaccharides. Can you remember? Hmm? We drew the structures of these as well, just representations, and we learned how the Disaccharides are formed out of monosaccharides. To how two monosaccharides combine in order to form a disaccharide. We discussed those things. Did you study as you promised me last week? Padan kalada? Did you study the structure? And we discussed about the three types of carbohydrates, their location, how do they occur, what are the other important facts. Is it? Turn on the videos, all of you. Balana, turn on the videos. Hi. So we have to start with the significance of carbohydrates. Is it? We have to start with the significance of carbohydrates. What are the importances of carbohydrates in living bodies? Right. So first of all, we can consider carbohydrates as an energy source. That is the most important function performed by those carbohydrates. They act as a source of energy, right? Acts as a source of energy. Now, due to the cellular respiration, actually carbohydrates produce energy. They undergo cellular respiration. You know, glucose is one of the main respiratory substrates. Oh, Windy, can you turn on the video, Pute? Right? Glucose acts as a main respiratory surface, right? Glucose reacts with oxygen. Releasing energy. Glucose reacts with oxygen, releasing energy, right? Then we call this as cellular respiration. So that is why we mention carbohydrates as a main source of energy. Carbohydrates as a main source of energy, right? Significance of carbohydrates. Significance of carbohydrates. Okay, then as an energy source, carbohydrates act as a primary and major source of energy for the functioning of cells. Glucose is produced by the hydrolysis of complex carbohydrates. Again, apeda kiwa starch. If we consider starch, so the monomer of starch is glucose. Right? Therefore, then starch is hydrolyzed into maltose. Then maltose is hydrolyzed into glucose. Right? Then glucose undergoes oxidation in order to produce energy. Right? Then as a storage compound, within brackets you can say cellular respiration. Within brackets you can say cellular respiration. Cellular respiration is the process by which carbohydrates release energy. Right? That happens inside the cells. So in the 6th unit we will learn about the cellular organelles. There you learn that mitochondrion is the powerhouse of a cell. So the cellular respiration happens inside the mitochondria. Then as a storage compound, in plants, what is the stored type of carbohydrate? Yes, what is the stored type of carbohydrate in plants? Hesandu, Rukshan, Tisas, Sasin, turn on the videos please. Yes. What is the stored type of carbohydrate in plants? Kyan balana?
Starch.
That is starch. Starch In plants. In animals, what is the stored carbohydrate? Yes? Murad. Glycogen. Glycogen in animals. Glycogen in animals. As a storage compound. Starch in plants and glycogen in animals. Hari. ඊළඟට as a structural component in plant cell wall. So what is the structural component available in the plant cell wall? මොනවාද තියෙන්නේ cell wall එකේ?
Cellulose.
Cellulose. What is cellulose? Cellulose කියන්නේ මොනවාද? It is a polysaccharide නේද? Cellulose is a polysaccharide. So as an example, you can say structural component in plant cell wall. Example, cellulose. It is a polysaccharide. Cellulose is a polysaccharide. Cellulose is a polysaccharide. Right? Hari. Method පුතේ cellular respiration. Shall we write the equation for cellular respiration? මෙතනම ලියලා තියාගමු the word equation of Indi and Tisas. Word equation for cellular respiration. I will write. So in cellular respiration what happens is glucose mainly reacts with oxygen, right, producing මොනවාද හැදෙන්නේ? In addition to energy, it produces, yes, carbon dioxide. That is what we exhale, right? Carbon dioxide. Then water vapor. Exhale, there contains water vapor as well. Once you exhale onto a glass or a mirror, you can see a misty appearance on it because we send water vapor as well. Carbon dioxide, water, actually water vapor, and the other thing is energy, what we need. So these are the products of cellular respiration plus energy. We call this as an oxidation reaction. Oxidation තමයි මෙතනදී වෙන්නේ. Reacting with oxygen. That is oxidation. Then right, then as a constituent of nucleic acid, then we are going to discuss about nucleic acids. The structure DNA and RNA. In DNA we find deoxyribose sugar. That is a 5-carbon sugar which we call as pentoses. Hari, the 5-carbon sugar which we call as pentoses. I will mention about it. Pentose sugar. In DNA we find deoxyribose. Deoxyribose is there in DNA. Then in RNA we find ribose sugar, right? RNA, that is ribose. And deoxyribose sugar. Pentose කියලා කියන්නේ 5-carbon sugar කියන එක. Then I have given main 4. ඊළඟට අපිට තව කියන්න පුළුවන් නම් you learned that the glucose synthesized during photosynthesis is translocated through the phloem tissue in the form of a sucrose solution. So sucrose is a disaccharide neda. As a transportation compound in plants as the fifth one. As a transportation compound compound in plants. As example, we can say sucrose. Sucrose getting translocated through the phloem tissue. Sucrose getting translocated through the phloem tissue. And finally, dietary fiber. We mentioned that in a balanced diet, in addition to the major nutrients and also vitamins and minerals, there should be fiber and water here, right? So dietary fiber is provided by cellulose, right? Enisa apita kiyanna puluwan providing dietary fiber. Then api ganna fruits, vegetables, in most of the plant materials, even in grains, we find this fiber, providing dietary fiber. As example, we can take cellulose. Api kendi kiyala kiyan, shaka kendi varga kiyala kiyan, cellulose. The stored type of carbohydrate in plants is starch. Fiber is cellulose. Plant cell wall is basically made out of cellulose. Hari neda? Sesandi and Thisas, please turn on the video. Finished? Balana Metuja, Menuli, Upeksha, Isumi, Janudi, Raika, Manuti, Tesini, Netumi, Amadi, Janot, Shenela, Limansa, Tichan, Deudini, Sasen, Ranu, Kumalcha, Denuri, Sesandi, Shaneli, Rukshan, Ovindi, Sachita. Sachita, is it your new first day? Are you a newcomer?
Yes.
Okay. Right. Cecily, you joined last week, I think, isn't it?
Yes, teacher.
Right. Then who else? Shaneli, right? Rukshan, Ovindi, Sachita, Tisas. Right, okay. Hurry, we'll move on. Tests to identify carbohydrates are another important thing. What are the tests now? When we are given a particular food item, we need to check whether it contains different types of carbohydrates, right? Different types of carbohydrates. So, first we will discuss about the starch test. Make a topic karamu mehama veda. We'll draw a table, right? You can draw a table with 3 columns. in the first column you can say the test. What is the name of the test, right? Then you have to say what is the method followed and the observations. For the method we will keep some more space, and then the observations, right? Observations. And here it is the method. Okay, so first we will mention about the starch test as number one. Number one, starch test. Starch test, right? Starch test. Have you all කරලා තියෙනවා starch test නේද? In grade 7 I think you have done the starch test, right? හරි. So here then අපි සාමාන්‍යයෙන් නම් මේක වෙන්න ඕනේ, normally we have to do this with an unknown sample of food to test whether it contains carbohydrates. දැන් මෙතන අපිට definitely we have to obtain results නේ. ඒ නිසා ඔයාට කියනවා starch තියෙන food item එකක්ම අරගෙන මේ test එක කරලා බලන්න කියලා. හරිද? I will show you videos as well. හරි. So you have to take a small amount of food, like you can take some rice, cooked rice, right? Or you can take a yam, a potato, like harida, and you have to mix it with water, grind it well and mix it with water. Then you can obtain an extract out of it. ඊට පස්සේ මොකද කරන්නේ? You can just put few drops of iodine solution. So what is the color of iodine solution? What the color in the iodine solution? Yes? Yes, Upeksha. What's the color of iodine?
Purple, right?
No.
Transparent.
No. Iodine solution එක මොන පාටයිද?
Brown color.
Yes, Amadi?
Brown color.
අපි ඒකට කියනවා yellowish brown කියලා. කහ පාටට හුරු දුඹුරු පාටක් තමයි තියෙන්නේ. Yellowish brown, right? හරි. ඔන්න method එක මම ලියන්න ඕනේ. You write down the steps. First step. ලියන්න. ඉවරයිනේ අඳලා table එක. Hi, first step, write like this. Take small amount of food. Take small amount of food. Take small amount of food and take small amount of food. Take small amount of food and mix with water. Mix with water. Mix with water. Add next step. ඊළඟ එක. Add a drop of iodine. 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 Hari. So what happens on the iodine wala color? Then you are asked to write a color change. වර්ණ විපර්යාසය ලියන්න කිව්වහම තිබුණේ මොන color එකද ඒක convert උනේ මොන color එකටද කියලා පැහැදිලිව කියන්න ඕනේ. Right? So yellowish brown color change එක තමයි yellowish brown. Yellowish brown turns to purplish blue, purple blue, නැත්නම් dark purple කියලා අපිට කියන්න පුළුවන්. In some of the books you find it as blackish blue. අපි කියමු dark purple කියලා. Dark purple. අද දම් පාටක් තමයි එන්නේ. නැත්නම් අපි කියනවා blackish blue කියලා. කළු පාටට මුළු නිල් පාටක්. Right? Dark purple. Now I will share the starch test for you to watch.
An important constituent of our daily food are carbohydrates, which we consume in the form of starch. Starch is a high-calorie food that converts to sugar during digestion. Starch is also produced by all green plants as an energy store. Today we will conduct an experiment to test the presence of starch in potato, rice, sugar, and salt. For this, we will need iodine solution, which is yellow-brown in color. Note the change in the color of iodine as it is put on the rice and potato. Both turn the iodine blue-black. Put some iodine on the sugar cubes. The color does not change. Salt too does not change its color. This proves that sugar and salt do not contain starch, while the starch in rice and potato reacts with iodine to form a blue-black compound. Now the reaction is because the blue-black color is basically the formation of polyiodide chains from the reaction of starch and iodine. And
Okay, Hari, that is all about the starch test. Then we will discuss about the test for monosaccharides. Hari, the test for monosaccharides. අපි මේකට කියනවා the test for reducing sugars කියලා. මම එදා කිව්වනේ the only non reducing sugar that we are discussing is sucrose කියලා. අපි මේකට කියමු test for reducing sugars. නැත්නම් we can say Eliana number 2. Test for monosaccharides. Test for monosaccharides and some disaccharides in Amaliana. I will write on my board. Liana, test for monosaccharides and some disaccharides. Hello? Hello?
Hello?
Oh?
Miss parcel එක order කරලා තියෙනවනේ online එකේ.
Oh?
මම මේ ගන්න කරන්ත පාර කොතනද?
බුද්ධ ජයන්ති මාවත.
ඒකේ කොතනද?
ඒකේ සුබ සාධන මාවතට හැරෙනවා.
සුබ සාතන මාවතේ කොතනද?
සුබ සාධන මාවතේ ටික දුරක් යනකොට අර වම් පැත්තේ පොල්වත්ත තියෙන එක පහ වුණාම දෙවෙනි ගේ.
අර සුදු පාට ලෝකයේද?
ඔව්.
හරි.
කීයට වගේ දෙන්නේ?
මිස් මම $1 පස්සේ එනවා.
හරි.
4290ක් තියෙනවා.
හරි.
Okay.
Test for monosaccharides and some disaccharides. Right, so within brackets you can say reducing sugar. Within brackets you can say reducing sugar. Right, reducing sugar. Reducing sugar. Reducing sugar. Hi. So what's the method? On the make ahagan. You have to take a solution of we'll say glucose. You are provided with a glucose solution. You can dissolve some glucose in water and obtain a glucose solution. Ranu, Rukshan, Methuja, Tesini. Then you have to add in the laboratory. There is the Benedict solution. නැත්නම් අපි මේකට ගොඩක් වෙලාවට කියන්නේ Benedict reagent කියලා. Right? You have to add Benedict. Benedict. මේක සමහර ඒවා Benedict's කියලාත් කියනවා. Right? Benedict's reagent or solution. Then you have to mix those two well by shaking the test tube. You can take a similar volume of each, right? If you take 2 cubic centimeters of the glucose solution, you can add glucose solution. Then you can add a similar volume of the Benedict solution or reagent. Benedict solution, right? 2 cubic centimeters of it is added into this. Then you mix the two components well. After that, you have to immerse the mixture in a hot water bath. Right? You have to immerse this in a hot water bath. You can keep a beaker on the tripod and you can provide heat by using a Bunsen burner, and this contains warm water, right? So Benedict's reagent is actually, it contains copper sulfate. What's the color of copper sulfate?
blue,
Blue. Then දැන් මොකද වෙන්නේ? ඔයා රත් කරනකොට ටික gradually the color changes. Blue becomes green, right? Blue green, ඊට පස්සේ yellow, අපිට කියන්න පුළුවන් green yellow කියලාත් අපි කියනවා, right? Blue green yellow, කහ පාටට උරු කොළ පාටක්, green yellow. Then it turns orange, and finally it becomes a brick red precipitate. අර ගඩොල් වල තියෙනවනේ අමුතු රතු පාටක් නේ, අන්නේ brick red precipitate. You know what a precipitate is? On the bottom of the test tube or the boiling tube, you will see a powder like precipitate is formed, brick red precipitate. So it shows a series of colors. Color changes gradually. Hari. අපි ලියමු එහෙනම් මතකද දැන්? You take a glucose solution, add Benedict solution, a similar amount mix, and immerse in a hot water bath. Then it will show a series of color changes like this. Hari. ලියමු එහෙනම් method. On a point form එකෙන් ලියන්න stepwise. Number 1, first one. Take a glucose solution. Within brackets 2 cubic centimeters. Sorry. Take a glucose solution within brackets 2 cubic centimeters. 2 cubic centimeters. Harida. 2 cubic centimeters. Then next point, add few drops of Benedict solution. Add few දැන් මම එන්නේ නෑ නංග, ඒක තමයි ඔයාලට කිව්වා 100 එක ගිහලා සෝදා ගන්න කියලා. මට මේක කරන්න බැහැනේ. Then add few drops of Benedict solution within brackets once again. You can write 2 cubic centimeters. 2 cubic centimeters. ඊට පස්සේ මොකද කරන්නේ? Immerse the above solution in a water bath and heat. Immerse the above solution in a water bath and heat. Immerse the above solution in a water bath and heat. Right? Hari. Now under the method, here we can just draw this. we can draw this. You have kept it on a tripod and there is a wire goes and you are providing heat by using a Bunsen burner. Okay, you can just show, right? So, and label, this is the test tube, this is the beaker, water, the tripod, and you have kept a wire mesh or a wire goes on the tripod, wire mesh or a wire goes, and you are using a Bunsen burner or you can use a sprit lamp to provide heat. You have to manage space. තව එක ටෙස්ට් එකක් තියනවා ලියන්න for test for sucrose. නිසා manage your space. Hi, finished? Then shall we write the observation? You can write the series of color change. Blue, green, ඔන්න ඔය order එක මතක තියාගන්න. Blue, green, yellow, orange, and finally a brick red precipitate. Hari ned? Shall I show you the video then? Balamu. Now this is the test I will share. අම්මගාව නැහැ මංගා ගාමගෙන නැහැ මෙතෙන්ට කතා කරන්න එපා මට හරි අමාරුයි මේ වැඩේ කරන්න.
Okay, food test for reducing sugars. You need a test tube rack, 2 test tubes, one for the control, one for the actual experiment. You need some glucose, 10 ml solution of glucose solution. You need 10 ml of water and what we call the Benedict solution. So you add 10 ml of water, you need 10 ml of glucose solution. You add a few drops of Benedict solution.
So you can see what they are doing. They added few drops of glucose and adding the Benedict solution. You can see the color
You
of
stir
Benedict.
it up.
Benedict has got
And
that
if
color
you want to
because
see
of the
the
presence
actual
of copper
experiment, you need to put
sulfate.
it in a water bath at temperature 70
And
degrees.
මොනවද ගත්තේ to the
So,
two test tubes?
You add
Hey,
10
බලන්න.
glucose solution, you need 10 ml of water,
හරි, water is added
and what
to
we call
both.
the Benedict
Then
solution.
Benedict solution.
You add 10 ml
10
of water,
milliliters of water as the control.
Yeah, 10
එකකට දාන්නේ
ml of
වතුර
glucose
විතරයි. To the other one he is adding 10 milliliters of
You
glucose
add a few drops
solution.
of
එකකට
Benedict
තියෙන වතුර
solution.
විතරයි. Then for both he is adding the Benedict solution. Control
You stir it a
setup.
bit,
මොකක්ද
and
control එකක් කියලා කියන්නේ? The setup which does not contain the factor that we are going to test. අපි test කරන්න යන දේ නැති කෙනා තමයි control එක කියලා කියන්නේ. අන්න ඕක අහලා තියෙනවා. Not only in this one but in other units also. There are photosynthesis වලදී මහාන what is the control කියලා. Control කියලා කියන්නේ the setup in which the factor that we are going to test is not available. අපි test කරන්න යන factor එක නැති කෙනා.
If you want to see the actual experiment, you need to put it in a water bath at temperature 70 degrees.
Then it is immersed in a hot water bath. May balana, the one with glucose, you can see that the color changes. In the other one, there is no change. On a color එක change වෙන හැටි බලන්න. ඔන්න දැන් අර orange වේගෙන යනවා. වේගෙන් color change වෙනවා. Then once you keep this for
So this
some
is a
time,
positive test.
it will form a
Here
brick
you are
red
in presence of
precipitate.
glucose.
පහළ
And here's your
මේක තව ටිකක් රත් කළා නම් ඒක ඇතුළේදීම you can see the brick red precipitate, right? Okay. That is the test for reducing sugar. Hari. ඊට පස්සේ ඔන්න අපි ලියමු the last test for carbohydrates, that is the test for non reducing sugar. අපිට sucrose කියලා කියන්න පුළුවන්. test for sucrose නැත්නම් we'll say non reducing sugar. hari මේකම තමයි කරන්නේ. But we are just doing a small difference. ඒ difference එකට හේතුව ඔයාලා මට කියලා දෙන්න ඕනේ. With the knowledge you have. හරිද? test for sucrose, or within brackets you can say non-reducing sugar, right? Non-reducing sugar. Non-reducing sugar. Test for non-reducing sugar. Hi. Then මෙතෙන්දි කරන්නේ you take on ඔයාලගේ textbook එකේ ඒගොල්ලෝ කියන you take 2 cubic centimeters of sucrose solution. නැත්නම් you can take sugar. The sugar dissolved in water. Sucrose solution. ඊට පස්සේ මොකද කරන්නේ? You add Benedict solution and immerse it in a hot water bath. But you don't observe any color change. මුකුත් වෙනසක් වෙන්නේ නැහැ. Right? After that you add few drops of an acid like මොනවාද ඔයගලන්ට දාන්න පුළුවන්, like sulfuric acid, dilute sulfuric acid, right? Few drops of dilute sulfuric acid. මෙහෙම add කරලා then you have added an acid නේ. So to neutralize it you can add few drops of sodium hydroxide as well in order to neutralize the acidic medium. It is not essential. Sodium hydroxide add කරන්න පුළුවන්. ඊට පස්සේ මොකද කරන්නේ? You have to immerse it in the hot water bath. You have to immerse it in the hot water bath. එතකොට ඔයාලා දකිනවා අන්න අර color change එකම ආපහු එනවා. දැන් මොකද්ද මේ acid එකක් add කරලා ඔයාලා කළේ? දැන් මට කියලා දෙන්න බලන්න. චුට්ටක් කල්පනා කරලා දැන් ඔයගොල්ලෝ දන්නා how sucrose is made. Sucrose කොහොමද හැදිලා තියෙන්නේ කියලා ගිය සතියේ කියලා දුන්නා. Then what why once we add an acid we obtain the same color change, and you know the test for monosaccharides. Ay mehmone. Any volunteer? Okay, Kian. Yes. Go. දැන් මතකද අපි කිව්වා sucrose how is it made? How is it made? How sucrose is made? Glucose කියන්න. මේ පාඩ පෙන්නන්නේ.
Plus fructose.
Glucose plus fructose. Glucose plus fructose makes sucrose. එතකොට glucose and fructose කියන්නේ මොනවද? What are they?
Monosaccharides.
Monosaccharides. This forms sucrose plus water. Sucrose plus water. And can you remember I told you this reaction is reversible. මේ පැත්තට අපි කියනවා condensation කියලා. Removal of water. Water molecule එකක් අයින් වුණාම මේ දෙන්නගේ sucrose හැදෙනවා. ඒකම අනිත් පැත්තට we call it as hydrolysis. Hydrolysis කියලා කියනවා කිව්වා නේද? That is known as hydrolysis. මේ පැත්තට condensation, මේ පැත්තට hydrolysis. එහෙනම් දැන් ඔයා acid එකක් add කරලා මොකද කරේ? In acids we have hydrogen ions. එතකොට hydrogen දුන්නා. sucrose වලට hydrogen දුන්නා. එතකොට මොකද උනේ? It is converted into glucose and fructose which are monosaccharides. එතකොට ඒගොල්ලෝ මේ Benedict's region එකට react කරනවා. තේරුණාද? කලින් acid එක add කරන්න කලින් color change එකක් නැහැ. හරිද? After adding the acid and immersing in a hot water bath, it showed the same color change. එහෙනම් ඔයා මොකද්ද මේ කරලා දීලා තියෙන්නේ? ඔන්න මතක තියාගන්න. By adding the acid you have hydrolyzed sucrose into glucose and fructose. එතකොට they give results for Benedict's solution. තේරුණාද? හවුද? Is that clear? The monosaccharides react කරනවා Benedict solution එකට. හැබැයි sucrose දුන්නේ නැහැ color change එකක්. Then we added an acid and heated it up. එතකොට it gave the same results. එන acid එකක් add කළාට පස්සේ මේ color change එක දෙනවා නම් that is sucrose. Hi, it is a non-reducing sugar. Clear?
Yes, teacher.
And instead name, write the name of the test. Then we'll write the method. එක ආයෙ අඳින්න ඕනේ නැහැ ඉස්සෙල්ලා ඇඳලානේ. එකම වෙනස we add a few drops of an acid. Take a sucrose solution. Take a sucrose solution. Within brackets say 2 cubic centimeters. 2 cubic centimeters. Right, into a test tube, into a test tube. Then next point, add few drops of Benedict solution. Add few drops of Benedict solution. Within brackets, once again 2 cubic centimeters. On the main step එක highlight කරලා use another color or after writing you can highlight or underline. මොකද්ද කියන්නේ? Add few drops of dilute sulfuric acid. Add few drops of dilute sulfuric acid. අපි ඒක මෙහෙම ලියනවා. මේ dilute sulfuric acid කියන්නේ කොහොමද මෙහෙම ලියනවා? DIL කියලා dot එකක් තියලා H2SO4 solution ने aqueous solution जलीय द्रावणයක්. ඒ නිසා මේ පහලින් within brackets you put AQ. අන්න ඔහම ලියනවා. මේවත් ඉගෙන ගන්න දැන්. හරිද මේ තුජ? DIL H2SO4 dilute sulfuric acid solution කියන එක තමයි මේ ලියලා තියෙන්නේ. Aqueous AQ for aqueous solution ජলীය द्रावණය. Hari. Add few drops of dilute sulfuric acid. Next point. Immerse in a hot water bath. Immerse in a hot water bath. Immerse in a hot water bath. Hi. Then you can write the series of color changes: blue, green, green yellow, orange and brick red precipitate. මේකේ පුතේ අර method එකට ඔයගොල්ලෝ within brackets දාගන්න ඔයගොල්ලෝ සමහර තැන්වල දකි then since we have added an acid in order to neutralize it they add few drops of sodium hydroxide aqueous solution as well. හරිද within brackets ලියා ගන්න to neutralize the medium. To neutralize the medium. Neutralize the medium. We can add few drops of sodium hydroxide solution as well. Sodium hydroxide solution as well. Sodium hydroxide solution. Sodium hydroxide solution. හරි. ඔන්න මම ඒකත් පෙන්නන්නම් the video.
Okay, here's the food test for non-reducing sugar. You need test tube rack, 3 test tubes, you need some cylinders, you need some, again, Benedict solution, hydrochloric acid, sodium hydroxide, and some sucrose solution. Okay, let's add 10 ml for our test control, the water. Then we have our sucrose solution, 10 ml.
They are adding water, sucrose, and what is the other one they added? On the sucrose. They mention I think something.
And we're going to add a few drops of hydrochloric
reducing
acid.
sugar they may have added.
And we'll test control. This one we don't put any hydrochloric acid, we'll leave it aside. We're going to take those 2 test tubes and we put it in the water bath for a few minutes. You put your 2 test tubes,
Hot water bath.
test control and the sucrose or sugar solution in the water bath for a few minutes. While they're cooling down, you have to neutralize the hydrochloric acid, so you need to add a few drops of sodium hydroxide, so it should be 10 drops.
Sodium hydroxide to neutralize the medium, acidic medium.
10. And once again, you don't touch that one here. Now it's time to add the Benedict solution. Add a few drops in each test tube. test tube, then we put them in the water bath for a few minutes. Okay, so we did the Benedict test with non-reducing sugar.
Now you
As you
can
see,
see
this
the
is a
color
control with
change
water.
in one only.
So,
that is
no
water.
reaction.
That is the
Here,
control
this is
setup.
the
Which
test tube
is
with sugar and
sucrose with
hypochloric
acid.
acid. So,
And
and
then
then you get
the
a
other
reaction
one,
because you broke down.
maybe
the
sucrose
saccharide
without
into
acid.
glucose
ඒක
and
ඒකට කිව්වේ නෑ sucrose
That
without
one
the
is
acid
basically
කියලා අපි
sugar,
ගමු.
so
එතකොට
sucrose.
හරි
with no
ඒක තමයි.
hypochloric
Sucrose without the acid. So without the acid it does not show any color change. හරි නේද? දැන් හොඳට හිතට ගියාද? Now I'm asked to mention about reducing and non-reducing sugars once again. Actually ඒක දැන් ඔයගලන්ට මෙතන explain කරන්න කියලා නැහැ reducing and non-reducing sugars. දැනට මතක තියාගන්න the only type of non-reducing sugar that we are discussing is sucrose කියලා. එතකොට sucrose මේ Benedict's test එකට react කරන්නේ නැහැ. මොකද අපි මෙතන chemical composition එක ගන්නෙ නැහැ නේ පුතේ. ඔයගොල්ලන්ට එච්චර දුරට ගන්නන්න කියලා නැහැ. නැත්නම් අපිට කියන්න පුළුවන් මේ color change එක වෙන්නේ what is the chemical compound formed ඒ ටික ඔක්කොම explain කරන්න පුළුවන්. ඒ ටික ඔයාලට වැඩි. ඒ නිසා ඔයාලගේ level එකේදී මතක තියාගන්න මොකක්ද? Sucrose තමයි අපි කතා කරන එකම non-reducing sugar. Right? So it does not give a direct result for the Benedict's test. Right? So once we add an acid and hydrolyze it, then it forms the respective monosaccharides, glucose and fructose. So they give results for the Benedict solution. අන්න ඒ ටික තේරුණා නම් හොඳටම ඇති. ඒ ටික පැහැදිලිද? හවුද? ඒක හරිනම් that's enough. Okay. මහන්සිද? නෑ නේද? මමයි කතා කළේ. That's the end of carbohydrates. ඊට පස්සේ ඔන්න අපිට කතා කරන්න තියෙනවා about proteins. හරිද? Proteins. But our next topic is about proteins. හරි proteins ගැන ඔච්චර දිගට කතා කරන්න නැහැ. හැබැයි තියෙනවා certain things we have. There is only one test for proteins. ඉක්මනින් කතා කරන්න පුළුවන්. දැන් චුට්ටක් මහන්සි නිසා අහගෙන ඉඳලා පැයක් water break එකක් ඕනේද? ඕනේ නැනේ. Do you want a water break? 2-minute water break. Right? දුවල ගිහලා වතුර ටිකක් බීලා එන්න. විනාඩි 2යි. ඊට පස්සේ I will share a video related to proteins. After that I am discussing. I'm starting the discussion about proteins. 2 minutes. Now it is 10:33. You have to come back by 10:35. Okay. දැන් ඔයා ක්ලාස් නේ? මොකද වාට දෙනවා මාරු වාට ගන්න මොකද? ඔය සතියක් විතර ටීවී එක ගලවලා තිබ්බනම් ඔයාලගේ ලෙඩ ඔක්කොම හොඳ වෙනවා නම් ඊටයි ඔයාටයි අක්කටයි. අක්කාද පොත් ගෙනින්නත් නැහැලු නේද ටීචර් එව්වා. චරසිහි නෑ. එයාට. ඔයාගේ පොත් ටික හරියද?
ඔක්කොම හරි කිව්වා.
ඒවට හිට කවර්ස් ටිකක් එහෙම දාලා ගන්න ඕනේ පිළිවෙලට. දැන් ඊළඟ එකනේ කඩවත කෑවද? කාලා ගියේ? ඔළුව රිදෙනවා නම් පැනඩෝල් එකක් දීලා යන්න. පැනඩෝල් තියෙනවා කබඩේ. මම උදේ පාන ඊළඟ යනවා මම. නෑ මම 12ට ඉවරලා කනවා. Okay, now the water break is over. Have you returned? Avane? Kobala Navanan, just raise your hand. Most of you have returned. Hi, then I will share the video related to proteins. Then make a good description එකක් තියෙනවා in the video, so just get an idea. දැන් මම මෙතන කියනවට වඩා දැන් පේනවානේ දකින්නකොට පැහැදිලි වෙනවානේ මේ protein එකක් කියන්නේ මොකක්ද කියලා නේද? Because none of us have seen a protein in the reality. So they will explain the structure, then we'll discuss.
Proteins play countless roles throughout the biological world. Some transport nutrients throughout the body. Some help chemical reactions to happen at faster rates. Others build the structures that make up living things. Despite this wide range of functions, all proteins are made out of the same 21 building blocks, called amino acids. Amino acids are made of carbon, oxygen, nitrogen, and hydrogen atoms, and some contain sulfur atoms. Selenocysteine is the only standard amino acid that contains a selenium atom. These atoms form an amino group, a carboxyl group, and a side chain, all attached to a central carbon atom. The side chain determines an amino acid's properties and is the only part that varies from amino acid to amino acid. Hydrophobic amino acids have carbon-rich side chains, which don't interact well with water. Hydrophilic, or polar, amino acids interact well with water. Charged amino acids interact with oppositely charged amino acids or with other molecules. Primary structure. The primary structure of a protein is the linear sequence of amino acids as encoded by DNA. The amino acids in a protein are joined by peptide bonds, which link the amino group of one amino acid to the carboxyl group of another. A water molecule is released each time a peptide bond is formed. This linked series of carbon, nitrogen, and oxygen atoms is the protein backbone. Protein chains often fold into two types of secondary structures, alpha helices and beta sheets. An alpha helix is a right-handed coil stabilized by hydrogen bonds between the amine and carboxyl groups of nearby amino acids. Beta sheets are formed when hydrogen bonds stabilize two or more adjacent strands of amino acids. The tertiary structure of a protein is the three-dimensional shape of the protein chain. This shape is determined by the characteristics of the amino acids making up the chain. Many proteins form globular shapes with hydrophobic side chains sheltered on the inside, away from surrounding water molecules. Membrane-bound proteins have hydrophobic amino acids clustered together on their exteriors, so that hydrophobic side chains can interact with the lipids in the membrane. Charged amino acids allow proteins to interact with molecules that have complementary charges. The functions of many proteins rely upon their three-dimensional shapes. For example, hemoglobin forms a pocket to hold heme, a small molecule with an iron atom in the center that binds to oxygen. quaternary structure. 2 or more polypeptide chains can come together to form one functional molecule with several subunits. The 4 subunits of hemoglobin cooperate so the complex can more easily pick up oxygen in the lungs and release it in the body. Different visual representations of proteins can give us visual clues about protein structure and function. This space-filling diagram shows all of the atoms that make up this protein. This representation, called a ribbon or cartoon diagram, shows the organization of the protein backbone and highlights alpha helices. This surface representation shows the areas in the protein that are accessible to water molecules. Most proteins are smaller than the wavelength of light. For example, the hemoglobin molecule is about 6.5 nanometers in size. Hemoglobin is found in high concentration in red blood cells. A typical red blood cell contains about 280 million hemoglobin molecules. The 3-dimensional shapes of proteins determine their function. The flexible arms of antibodies protect us from disease by recognizing and binding to pathogens and targeting them for destruction by the immune system. The hormone insulin is a small, stable protein that can easily maintain its shape while traveling through the blood to regulate blood glucose level. Alpha amylase is an enzyme that begins digestion of starches in our saliva. The calcium pump is aided by magnesium and powered by ATP to move calcium ions back to the sarcoplasmic reticulum after each muscle contraction. Ferritin is a spherical protein with channels that allow iron atoms to enter and exit depending upon an organism's needs. On the inside, ferritin forms a hollow space with iron atoms attached to the inner wall. Ferritin stores iron in a non-toxic form. Collagen forms a strong triple helix that is used throughout the body for structural support. Collagen molecules can form elongated fibrils, which aggregate to form collagen fibers. This type of collagen is abundant in skin and tendons. Learn more about the functions and 3D structures of proteins and other molecular machines at the RCSB Protein Databank.
Okay, so you saw the structure of proteins. Actually, it is not that a simple structure, right? Actually, this is a polymerized molecule, right? Turn on the videos. First of all, if we consider the elements present in a protein, in the carbohydrates, we mentioned that they contain carbon, hydrogen and oxygen. Right? In the case of proteins, proteins is an essential constituent in all living cells. So what are always present in proteins? Carbon. I will use another color. Right? It contains carbon, carbon, hydrogen, oxygen and nitrogen are also available always in proteins. CHON are available always. And in some of the proteins you can find sulfur as well. Sulfur, it's capital S. That can also be present in some of the proteins. Even in the oral paper they had asked what are the elements present in proteins. So you have to say carbon, hydrogen, oxygen, nitrogen essentially, and sometimes they contain sulfur in some of the proteins. Harida, Limansa, Ovindi, Ranu. Hari. 17% of the mature human body is composed of proteins. If we consider the dry mass, 17% of it is made out of proteins. So proteins is a complex molecule made up of polymerized monomolecules, the amino acid molecules. දැන් මතකද අපි polysaccharide එක හැදිච්ච හැටි ඇඳලා පෙන්නන monomers වලින්. අන්න ඒ වගේ proteins are made out of polymerized amino acid. සමහර amino acid කියලා කියනවා amino acid molecules. Right? So amino acids are the basic units from which proteins are made. අන්න පුංචිම unit එක තමයි amino acid, right? In the case of proteins. Hari. එතකොට how many such amino acids? There are 20 such amino acids. මේ වගේ amino acids 20ක් තියෙනවා. And mainly contributed for the formation of proteins. Then there are 20 amino acids, but there are several hundreds or maybe thousands of proteins. ඒ කොහොමද හැම වෙන්නේ? දැන් හිතන්න in the English alphabet there are 26 letters නේ. Okay, you write the first 20 letters කියලා හිතන්න. එතකොට මේ ලියාගෙන යනවා. A B C D normally the first 20 letters. ඊළඟට බලන්න how many methods that you can write these 20 letters in different sequences. හිතා ගන්න පුළුවන්. මේවා order එක මාරු කර කර මේ 20 ආයි ලියනවා. කොච්චර ලියන්න පුළුවන්ද? නේ? හිතා ගන්නත් අමාරු නේද කොච්චර ලියන්න පුළුවන්ද කියලා. අන්න ඒ වගේ තමයි. These 20 amino acids combine in different orders and form large number of proteins. ඒ වගේ ඒගොල්ලෝ මාරුවේ එක එක sequence වලින් combine වෙලා they form different proteins. Okay? අන්න එහෙමයි proteins ගොඩක් හැදෙන්නේ amino acids 20ක් තිබුණාට. Though there are 20 amino acids, large number of proteins are formed like that. Hari. Then these are for extra knowledge. Plants are able to make amino acids they need. But animals are unable to synthesize all amino acids we need. We can't synthesize all the amino acids we need inside the body, but plants can. Right? Therefore, they must obtain some ready-made amino acids directly from food. They are termed as essential amino acids. Then, now the vegetarians, they have to obtain the essential amino acids from food. Right? Because since they don't consume any animal proteins, since they don't consume any animal proteins, they have to obtain them from certain food items, plant food items, plant food materials. So there are protein rich food, tofu, even mushrooms contain a lot of proteins. So tofu is made out of soya protein, soya milk, right? They are rich sources of proteins. ඔගොල්ලෝ කැමති නැති වුණාට ඉතර රස නැහැ කියලා කැමති නැහැ tofu වලට නේද but it is a rich source of protein. ඊළඟට පුතේ මේතන පෙන්නලා තියෙනවා the structure of a typical amino acid molecule. මොකද්ද typical කියන්නේ? Typical is the common structure. හරිද? common. මේ බලන්න there is a center carbon atom. මේ මැද ඉන්නවා carbon එකනේ. center carbon atom. Right. ඊළඟට You know carbon can make 4 bonds. එතකොට මේ bonds හතරේ කවුද ඉන්නේ? එක්කෙනෙක් තමයි hydrogen. Right? There is a hydrogen atom. ඊට පස්සේ අපි මෙයාට කියනවා carboxyl group. COOH carboxyl group is there. ඊළඟට ඉන්න amino group. අපි amino group එක NH2 කියලා ලියනවා තමයි. හැබැයි මතක තියාගන්න කාබන් වලට attach වෙන්නේ මේ දෙන්නගේ nitrogen. එච්චර විස්තර ඕනේ නැහැ. ඒ උනාට දැනගෙන ඉන්න මේ NH2 කියලා මෙහෙම ලිව්වට කාබන්ට attach වෙන්නේ nitrogen වලින්, right? ඊට පස්සේ මෙන්න මේ R group R කියන එකනා තමයි in the 20 amino acids they differ. හරිද මේ R කියන group එක එකනා වෙන්න පුළුවන්. For example, for the simplest amino acid glycine there is a hydrogen atom instead of R. On a glycine වල hydrogen තියෙනවා R විදියට. තව එකක CH3 කියලා group එකක් තියෙන්න පුළුවන් as R. එතකොට මේ R කියන group එක වෙනස් වෙලා තමයි there are 20 different amino acids. ඒ ඇති. හරිද දැන් මේක තියෙනවා දැන් අපි මේ protein structure එක ගත්තොත් ඔයාලා අර වීඩියෝ එකේ දැක ගොඩාක් deep කතා කරන්න පුළුවන්. හැබැයි අපි දැනගන්න ඕනේ we are restricted to a syllabus. We are given a teacher's guide, there is a textbook, and from those only they will ask questions. ඒ නිසා ඔයගොල්ලන්ට protein structure එක deep උගන්වලා මම මෙතන වෙලාව ගන්නවා කියන්නේ අපරාධය. මොකද ඔයාලගේ ultimate objective is facing the O level exam and scoring good marks for that. නේද? එතකොට අපි මේ paper එකට දැන් කවදාවත් මේකේ deep structure එක අහලා නැහැ. මේ for extra knowledge කියලා දීලා තියෙන part එක they don't ask for the O level exam. සමහර term test papers වල එහෙම මේක අහලා තියෙනවා. හැබැයි in the O level paper they will not ask. ඒ නිසා මම deep යන්නේ නැහැ. මේ surface knowledge එක දෙනවා. ඔයගොල්ලන්ට exam එක target කරලා. නැත්තම් biochemistry කියලා අපි ඉගෙනගෙන තියෙන ඔක්කොම මෙතන කියන්න ගියොත් it is a waste of time. Waste කියලා කියන්නේ දැනුම ගන්න එක wastage එකක් නෙවෙයි. හැබැයි we have to manage our time නේ for the other lessons also. නේ? Hi. R represents a group containing both carbon and hydrogen. Due to the change in R group, 20 amino acids are present. All proteins present in organisms from bacteria to humans are made up of those 20 amino acids, 20 different amino acids which have combined in different sequences. Then here it says, the simplest amino acid is glycine. Here hydrogen is present as the R group. I mentioned those. ඊට පස්සේ බලමු how amino acids combine in different sequences to form different proteins. Hari. So these 20 amino acids, as I told you, they combine. They combine in different sequences in order to form different proteins. මෙතන ලියන්න amino acids combine in different sequences to form different proteins. හරි. එතකොට අපි මේ amino acid එකක් ගත්තොත් if we just indicate an amino acid like this. This is one amino acid. This is another amino acid. These two are combined by a bond known as a peptide bond. මෙන්න මෙතන මැද තියෙනවා peptide කියලා bond එකක්. Right? Peptide bond. Peptide bond. This is the peptide bond. හරිද? ඔය විදිහට number of amino acids combine with peptide bonds and they make a polypeptide chain. There is a polypeptide chain එකක් හදනවා. One amino acid, another amino acid, then don't have any other color, another amino acid. Likewise, they combine with large number of amino acids and form a polypeptide chain like this. Poly කියන්නේ many කියලා ඉදා කියලා දුන්නා නේද? A protein may possess one or more peptide chains. මේ වගේ peptide chains එකක් හරි කීපයක් තියෙන්න පුළුවන් in a protein. In the video you saw there is a primary structure, secondary structure, tertiary structure. තව ගියාම තියෙනවා coordinate structure කියලා. එහෙම there are different structures in proteins. In A level biology you will learn. Chemistry වල නෙමෙයි biology වලට කියලා දෙනවා first unit එකේ. දැනට තියෙනවා. ඊට පස්සේ that's enough. A protein may possess one or more peptide chains. That's all. ඊට පස්සේ name some protein rich food. මොනවාද protein rich food? කියන්න බලන්න. Yes. Yes, so Indy. What are the protein rich food? Kishan. මොනවාද protein rich food කියන්නකෝ? Meat. Oh. Fish.
Eggs.
Eggs වල මොකක්ද? Eggs වල there are two parts නේ. Egg white contains proteins. හරිද? Egg white contains proteins. Egg yolk එකේ වැඩිපුර තියෙන්නේ lipids. The yellow part contain lipids.
Chicken breast.
Chicken breast හරි අපි meat කියලා common ලියලා තියෙනවානේ. ඊළඟට processed meat items tofu is made out of මොනවාද soya milk tofu mushrooms ඊළඟට you can even say dried fish can also be taken ඊළඟට dairy products yes dairy products like yogurt නේද ඊළඟට plant materials මොනවාද tofu hari plant material එකක් ඊළඟට beans lentils then dal beans dal ඒ වගේ අපි lentils කියලා කියනවා හරිද lentils ඊළඟට nuts and seeds
White cereals.
මොනවද පුතේ?
White cereals.
Cereals හරි cereals. එතකොට ඔයගොල්ලෝ කිව්වේ මොනවද? Cheese, yogurt they also contain. Cereals හරි. These are rich in protein. So if there are vegetarians they have a certain plant-based proteins neither they have to pay more attention since they are not taking animal proteins they have to take a number of plant-based proteins Harine finished Hirun is it your first day
Yes, teacher.
right Hi State the importance of proteins in living bodies. අන්න අපි ඉක්මනින් ආවනේ importance of proteins. එතකොට මොනවාද? So proteins are also important as an energy source, but remember carbohydrates act as the main energy source. හරිද proteins ඊළඟට lipids release more energy than carbohydrate oxidation. එහෙම එකකුත් තියෙනවා. එතකොට when carbohydrates and lipids are not adequate they make use of proteins as well to produce energy හරිද ජනෝත් මොකද දුකෙන් ඉන්නේ ජනෝත් right hari okay එහෙනම් ලියන්න number 1 as an energy source number 1 Tisas, where are you? Hari. So under that you can say when the energy supply from occasionally lipids and carbohydrates is not enough. Lipids and carbohydrates is not enough. When the energy supply from lipids and carbohydrates is not enough, proteins also act as a source of energy. Proteins also act as a source of energy. Proteins also act as a source of energy. Hari. Then number 2. To make structural compounds, not structural components. Number 2. To make structural components. Structural components. Structure එක හදන්න කොහොමද proteins contribute කරලා තියෙන්නේ? Now you know there is a cell membrane in both plant and animal cells නේද? Structural components. Cell membrane. So cell membrane is made out of cell membrane එකේ තියෙන එක එක දේවල්. Right? There are phospholipids තියෙනවා. අන්න එහෙනම් lipids උත් contribute වෙලා තියෙනවා structures හදන්න. Phospholipids කියලා જાතියක් තියෙනවා. ඊට පස්සේ there are glycoproteins. අන්න proteins are. Right? And there are cholesterol molecules as well. Cholesterol, it coming under lipids. Hari, එහෙනම් cell membrane consists of glycoprotein. Glycoproteins. Cell membrane consists of glycoproteins. හරිද? ඊට පස්සේ now in our hair as well as on nails we find මොනවාද? keratin. ඊළඟට in birds feathers we find keratin. They don't get wet. They get wet means they don't absorb water. දැන් අපි නෑවහම does hair absorb water? වතුර උරා ගන්නවාද යා? Nails, feathers of birds ඒගොල්ලෝ තෙමුනහම අර මෙහෙම ගැස්සුවාට පස්සේ the wetness is no more there නේද? නැත්නම් ඒගොල්ලන්ට වැස්සට තෙමුනහම පියාඹන්න බෑ. අය අර වතුර cotton wool වගේ වතුර උරා ගත්තා නම් නේ they won't be able to fly after the rain නේ. එහෙනම් keratin in මොනවාද? in hair, nails and feathers of birds, right? They have keratin. It's a protein. Keratin. Hari. ඊට පස්සේ තව මොනවාද? Then අපේ තියෙනවා in our skin, just under the skin actually, collagen. Collagen is also a protein that is available in our connective tissues. හරිද? එතකොට when on aging the elasticity of this collagen fiber is lost. හරිද? අර elastic කියලා කියන්නේ අපි rubber එකක් ඇද්දාම නැත්නම් elastic එකක් ඇද්දාම once we release it, it goes to the original position. අපෝ හිටිය විදිහටම හැදෙනවා නේ. But when the elasticity is lost, ඇදුනහම ඇදිලාම ඉන්නවා. Right? That is why on aging we find the wrinkles on the skin. අර හැම රැලි වැටෙන්නේ the elasticity of the collagen fiber is lost. Hari. So in connective tissues of animals. Connective tissues of animals, we find collagen. Collagen is a protein. Likewise, there are so many different types of proteins which make the structure in both plant and animal bodies. Hari, ඊට පස්සේ තව මොනවාද? Then අපේ තියෙනවා in our skin just under the skin actually, connective tissues of animals, we find collagen. Collagen is a protein. Hari, there are so many different types of proteins which make the structure in both plant and animal bodies. That is the second one. Now the number 3. All enzymes. අපි enzymes ගැන ඊළඟට කතා කරනවා. All enzymes are proteins. All. හොඳට මතක තියාගන්න. All enzymes are proteins. All enzymes are proteins. මොනවාද enzymes කියන්නේ? What's the use of enzymes? මොකටද අපිට enzymes ඕනේ වෙන්නේ? Sesandi. Yes. For what do we need enzymes? Yes.
For biochemical reactions.
For biochemical reactions. For example, then we got the digestion, food digestion inside our digestive tract, right? So a series of biochemical reactions take place. Then when we are doing a chemical reaction in the laboratory, sometimes in order to speed up the reaction, we will need to heat it up. But inside the body, those are not possible. So several hundreds of biochemical reactions are happening in the body and they need to be speeded up. So accelerate the reactions, speed up the chemical reactions. And for that we need enzymes, right? So in grade 11, 6th unit, you are learning about the structure of the human digestive system, not only the structure but also the functioning. So enzymes ගැන කියනවා. හරිද? So there are a lot of enzymes secreted in the body. අපි මේවට කියනවා biochemical catalysts කියලා. ඇයි? Bio means in living bodies, right? Chemical substances which speed up the rate of chemical reactions. Biochemical catalysts කියලා කියනවා. හරි නේද? එහෙනම් all enzymes are proteins කියලා ලියුවා. ඊට පස්සේ you can mention that. Under that you can say all biochemical reactions take place in organisms are catalyzed by enzymes. In number 4 we can say, fourth one we can say as hormones. On all hormones are proteins as hormones, because there are hormones with a protein origin as well as lipid origin. Hormones proteins වෙන්නත් පුළුවන්, lipids වෙන්නත් පුළුවන්. Enzymes all are proteins, right? එතකොට hormones මොකද කරන්නේ? දැන් අපි ගත්තොත් growth hormone, human growth hormone, GH කියලා කියනවා. Growth hormone, that is important for the growth of our body, right? ඊළඟට insulin is a hormone which is secreted in order to control our blood glucose level. Right, when there is glucose in our blood in excess, that glucose is converted to some other form, glycogen, and stored in our body. That is how our blood glucose concentration is regulated. But in diabetic patients, this does not happen due to many reasons. So they are given insulin externally, sometimes injected, sometimes as tablets. They have to take it, insulin, right? එතකොට එයා කරන්නේ එහෙනම් මොකක්ද මේ අපි මේකට කියනවා ඇයි අපි blood glucose level එක maintain කරන්නේ? Everything in our body should be maintained at an optimum level. අපි ඒකට කියනවා homeostasis කියලා. right? Once again in grade 11 you will learn homeostasis. We have to maintain the internal environment of our body at an optimum level, right? That is what we call as homeostasis, right? Homeostasis. And for coordination of different things we need hormones, and under hormones you can write hormones involved in homeostasis and coordination of organisms. Coordination of organisms. And then you can give examples: insulin and growth hormone. හරි. ඊට පස්සේ අපිට කියන්න පුළුවන් as antibodies ඒ මොනවාද? antibodies as antibodies ඒ කියන්නේ in සිංහල අපි එකට කියන ප්‍රතිදේහ කියලා. Antibodies. What is the importance of an antibody? So they are fighting against the pathogens that enter our body. They protect our body from pathogenic microorganisms. Then ඔයගොල්ලෝ අහලා තියෙනවානේ there are certain diseases. Once we get the disease, we will not get it again in our lifetime. කියන්න බලන්න example එකක්. Chicken pox. Yes, Shenela. එතකොට once we get chicken pox once, we will not get it again. The reason is chicken pox is a viral disease. On a virus entered our body, right? The virus has entered the body. එතකොට මොකද වෙන්නේ? දැන් අපිට තියනවානේ we have the immune system, right? So from our immune system, actually the white blood cells contribute for this. They develop antibodies. ඔන්න දැන් antibodies හදාගත්තා. හරිද? Now there are antibodies in our body which will act against that particular virus. ඒ virus එකට එරෙහිව හදාගෙන තියෙන antibodies ටික. හරිද? දැන් ඔන්න the disease is cured now. Then we don't have the pathogen in the body, but the antibodies are there, right? හදාගත්ත antibodies ටික එමම තියෙනවා. ඊට පස්සේ මොකද වෙන්නේ? Then the virus is always there in the environment නේ. virus එක environment එකෙන් අයින් වෙලා නැහැ. So in the second time also it will try to enter the body. දැන් මොකද කරන්නේ? There is a force. නේද? හමුදාවක්ම ඉන්නවා ඇතුලේ to fight against the virus. In the second time when the pathogen enters, the same virus enters. එයා විතරයි ඒ virus එකට තමයි මේ antibodies හැදිලා තියෙන්නේ. Right? So they will fight against that and destroy. So we will not get the disease once again. ඩෙන්ගි වලත් එහෙම වෙනවා. හැබැයි ඕගොල්ලෝ අහලා ඇති ඩෙන්ගි දෙතුන් සැරේ හැදිච්ච අයත් ඉන්නවා. ඒකට හේතුව තමයි ඩෙන්ගි is caused by different viral types. හරිද? එතකොට ඒකේ තියෙන ඩෙන්ගි වල there are different variants. ඒ නිසා එක variant එකක් ආපහු හැදෙන්නේ නැහැ. හැබැයි අනිත් variant එකෙන් ඩෙන්ගි හැදෙන්න පුළුවන්. And එහෙමයි වෙන්නේ. So these are the antibodies. So remember they are there to protect our body against the pathogens. Pathogens කියන්නේ disease causing agents නැත්නම් microorganisms. Okay. හරි. එහෙනම් අපි ලියමු under the as antibodies you can say they are produced in the body to protect from microorganisms. To protect from pathogenic microorganisms. To protect from pathogenic microorganisms entering the body. Pathogenic microorganisms entering the body. Hi. ඔයාලා පස්සේ ඉගෙන ගන්නවා මෙතනත් ඕනනම් අපි ලියලා තියාගමු මේ the white blood cells provide immunity from two different ways. Immunity from white blood cells. ಇದು ಮಾಡಿ. Immunity ඔයාලට ඉඳ තියනවා නම් ලියා ගන්න. ඉඩක් හොයාගෙන ලියා ගන්න. Immunity by white blood cells. කොහොමද දෙන්නේ? වර්ග දෙකයි. එකක් තමයි producing antibodies. They produce antibodies. And the other method is known as phagocytosis. Phagocytosis. What is that? Then හිතන්නකෝ this is the white blood cell කියලා. හරිද? Then a pathogen has entered into the body. දැන් මොකද කරන්නේ? This white blood cell engulf the pathogen. හරිද? නෑ එයාගේ ඇතුළට අරගෙන මෙයාව digest කරලා දානවා. Right? Phagocytosis. So within brackets engulf the pathogens and destroy. Engulf the pathogens and destroy. හරිද මේක එක සැරයක් අහුවා මේක අපි grade 11 unit 6 වල කියනවා blood circulatory system එකේදී in the O level paper they are asked to answer this. ඒ නිසා දැන්ම මතක තියාගත්තට කමක් නැහැ. These are the two methods how white blood cells protect our body from diseases. Phagocytosis and producing antibodies. Antibodies ගැන මෙතන කිව්ව නිසායි මම ඒකත් මෙතෙන්ට include කරගන්න කියලා කිව්වේ. පැත්තකින් ලියා ගන්න. මේක protein වල function එකක් නෙවෙයි. Phagocytosis කියන්නේ. හරි නේද?
Next.
Yes, Chaneli?
Means only producing antibodies is the function of protein, right?
Yes. හරි. ඊළඟට තව එකක් අපිට කියන්න පුළුවන්. We can mention one more. හරි. අනිත් දේ තමයි බද්ද දන්නවද? Act as respiratory pigments. හරි මේක ලියලා ඉවර නෑ නේද? ලියන්න pigment. Hello?
Hello, it's from your TV searching.
Oh.
Parcel as you know, you can know that.
Oh.
Then hardly you will understand. I mean, part of our meeting.
Oh.
Part of our
ඇවිල්ලා සුබසාධන මාවත කියලා තියෙනවා. ඒකේ ටික දුරක් යනකොට අර වම් පැත්තේ පොල් අත්තක් තියෙන්නේ අන්න ඒක පවුනාම තියෙන දෙවනිගේ. ඔව් අර කළු රෝලර් ගේට් එකක් තියෙන්නේ. කීයට වගේ දෙන්නේ?
සාමාන්‍ය 1.42 අතර වගේනවා.
හරි.
මම ඇවිල්ලාම කෝල් දෙන්න මාකර් තියගත්තා.
හරි මේ ඇවිල්ලා බෙල් එක ගහන්නකෝ. Hari. The last one we can say මොකද්ද? Producing නෙමෙයි respiratory pigments. Yes, as a component of respiratory pigments. Shall I clean? Respiratory pigments as respiratory pigments. මොකද්ද අපිට තියෙන respiratory pigment එක? ශ්වසන වර්ණකය මොකක්ද අපේ? What is it? That is hemoglobin. Our blood is red due to the presence of hemoglobin. The red color is given because of that. Hemoglobin. Right. Hemoglobin. Due to the presence of hemoglobin. hemoglobin වල පුතේ මතක තියාගන්න මේ heme කියලා කියන්නේ දැන් ඔයගොල්ලෝ අහලා ඇති once you have iron deficient වුණාම once you go to the doctor he observe the lower part of your eye the nails if they are pale අර නිකන් සුදු මැලි වෙලා නම් they guess that you have iron deficiency because the reddish color is given by hemoglobin in hemoglobin this heme කියලා කියන්නේ iron හරිද globin කියලා කියන්නේ it is a protein component right එතකොට hemoglobin මොකද කරන්නේ transports oxygen in red blood cells හරිද in red blood cells එතකොට ඇත්තටම වෙන්නේ මේ hemoglobin නම් if this is the hemoglobin molecule it combines with 4 oxygen molecules හතර පැත්තෙන් නෙමෙයි 4 oxygen molecules and combine with 4 oxygen molecules on a hemoglobin oxygen කියන්නේ O2 නේ එතකොට හතරක් වුණාම O8 වෙනවා HbO8 කියන විදිහට තමයි මේ ඔක්සිජන් ට්‍රාන්ස්පෝර්ට් කරන්නේ මේකත් ඇඩිෂනල් දැන් මේවා කියද්දි අර ගොඩක් දේවල් තියෙන නිසා කියවෙනවා හැබැයි ඉතින් knowledge එකට හොඳයි නිකන් ලියා ගන්න right heme is iron globin is protein එතකොට in day to day lives අපි දන්නවා iron deficiency if we are iron deficient hemoglobin production is affected එතකොට hemoglobin අඩු වෙනවා කියන්නේ oxygen transportation is affected oxygen නැති වෙනවා කියන්නේ මොකක්ද cellular respiration is affected. Cellular respiration is the process by which we get energy. එතකොට energy අඩුයි කියන්නේ we are very lethargic නේද? වාඩි වුණාමයි නේ? හරි කම්මැලි lethargic නේ? inactive hypoactive හරිද? එහෙම වෙන්නේ ඔන්න iron deficiency නිසා කියලා දන්නවා. එහෙනම් ඔයාලට එහෙම දැනෙනවා නම් I am very lethargic කියලා you have to check whether you have iron deficiency hemoglobin අඩුයිද කියලා once you take a full blood count you will be able to see the amount of hemoglobin available හරිද දැන් එහෙම අවස්ථා තියෙනවා සමහර වෙලාවට do you get enough food sometimes due to a worm impact then if there are worms inside the digestive tract they may be obtaining more blood ඒ වගේ ප්‍රශ්න තියෙනවා එක එක වෙලාවට ඒ නිසා you have to be on alert. Hari, that's all. About the importance of proteins in living bodies. Finally, a test to be done in order to identify proteins. Body test එකක් තියෙනවා. මොකක්ද කරන්නේ? You have to obtain a solution containing proteins. Right, solution containing proteins. Mama gannawa. I can take egg yolk. Egg yolk name egg white. Natham or a dal extract. Kohomada dal extract එක ගන්නෙ? You can put some dal into the mortar, and you, by using the pestle, you can just grind that at school in the laboratory and mix it with water. Right? Hondata grind karala, mix it with water and obtain dal extract. Right. And a protein containing solution is there, liquid is there. ඊට පස්සේ I have to add ඔන්න වර්ග දෙකක් add කරන්න තියෙනවා. මේකට මම දානවා number 1 an extra volume of sodium hydroxide solution NaOH. දැන් දානවා aqueous solution කියන්නේ මෙන්න මෙහෙමයි කියලා AQ, an extra volume of sodium hydroxide. ඒ කියන්නේ මෙයාගේ volume එකට සමානව වගේ. right? ඊට පස්සේ දානවා few drops of බින්දු කීපයක්, few drops of copper sulfate solution. Copper sulfate solution. CuSO4AQ. Copper sulfate solution. හරිද? ඔම දාලා once you shake කරන්න ඕනෙත් නැහැ සමහර වෙලාවට, you will be able to see the color change. මොකක්ද color change එක? Pinkish purple color එකක් you will be able to see. හරිද? Pinkish purple. ලියමුද? Write down the steps or stars dala hari kumari, you can write the steps. On the first step you can say obtain a solution containing protein into a test tube. Obtain a solution containing protein into a test tube. Next step. Add an extra amount of sodium hydroxide solution. Add an extra amount of sodium hydroxide solution.
Can you repeat?
Add an extra amount of sodium hydroxide solution. Last step. Add few drops of copper sulfate solution. Few drops of copper sulfate solution. Hi. Observation. Observation. The mixture turns pinkish purple color. Pinkish purple. Pinkish purple. මෙන්න මේ ටෙස්ට් එකට අපි දෙනවා නම් මොකක්ද? Biurate test. සමහර අය මේක biurate test කියලා කියනවා. Biurate නෙමෙයි ඇත්තටම. This is the biurate test. Right? Biurate test. I will show you the biurate test, how it is done. I will share. When I am sharing the screen, අර පැත්තකින් ඔයාලට කොටුවක් පේනවාද my screen? ඒ කියන්නේ මගේ මේ ස්මාර්ට් බෝඩ් එක කොටුව පේනවාද වෙනම? No, I just wanted to check whether it is a disturbance for you. Manuti, mage screen එක share කළාම on your right hand top corner can you see another rectangle?
Yes, teach.
I will try to remove that balloon. මට ඒක පෙන්වන්න නැහැ ඒකයිද I have to remove it but it is not visible for me. So they are doing it for different solutions. Then sugar solution එකට කළාට result එකක් තියෙයිද කියලා ඔයාලා හිතලා බලන්න. If it is blue, if the result is blue, there is no any change, negative. If it is purple only, it is a positive test. On a milk වලට දානවා. Sodium hydroxide and few drops of copper sulfate. On a positive neither. That's a positive test. Purple. When we are just comparing the colors, observing color changes, it's the best to use a white background. Okay, Manuti. We have to use a white background. Then මෙතන white background එකක් නෙමෙයි අරන් තියෙන්නේ. Color change එකක් පෙන්වන්න හොඳම white background එකක්. Taken in the laboratory, sometimes we use a white porcelain tile කියලා කියන්නේ. මොකද negative or positive, that's a positive result නේ are turning to purple gradually. Egg white should be positive, must be positive. Peanuts මොනවද? Peanut extract. What do you think? Should be positive. So we mentioned the nuts and seeds under the protein containing substances. Hi. That's all about the biurate test. Okay. Then we have to talk about enzymes. That is also under proteins. All enzymes are proteins කියලා කිව්වනේ. හරි. අපි බලමු පුංචි වීඩියෝ එකක් related to enzymes.
Enzymes. human body is a product of different chemical reactions and processes. But what controls these reactions? In 1833, a French chemist, Anselme Pierre, was the first to discover the vital force that drove these reactions and named it enzyme. Enzymes are substances, proteins, or in some cases, ribonucleic acid, RNA, that speed up a biochemical reaction by modifying specific substances called substrates. Enzymes are supremely selective in who they bind to and modify, hence their specificity. Mode of action. Easier said, but how does a tiny enzyme speed up a chemical reaction? The answer to this is simple. Enzymes perform this critical task by lowering a reaction's activation energy, that is, the amount of energy needed for the reaction to begin. Enzymes bind to their substrates, hold them in such that chemical bond breaking and bond forming processes take place more easily. Enzymes have a specific place in them called active site, where the substrate binds and real-time action takes place. Active site has a specific size, shape, and chemical behavior rendered to it by specific arrangement of amino acids. Thanks to these amino acids, an enzyme's active site is unique only to a particular substrate. In addition to the active site, many enzymes consist of a non-protein part called cofactor. Cofactors may be cations, positively charged metal ions, activators, bound temporarily to the active site to activate the enzyme. Organic molecules, vitamins or vitamin products, coenzymes that join enzyme-substrate complex temporarily. Prosthetic groups, permanently enzyme-bound. Many enzymes only perform their catalytic role when associated a coenzyme. An inactivated enzyme, protein, along with its coenzyme, non-protein, makes up a system called holoenzyme. To add to the complexity, scientists term this inactivated enzyme as apoenzyme. And therefore, the equation becomes holoenzyme equals apoenzyme plus coenzyme. Models of enzyme action. Lock and key hypothesis. This is the simplest model to represent how an enzyme works. The substrate simply fits into the active site to form a reaction intermediate. Just like the key fits in its specific lock, the shape isn't changed here. Rather, the structure of the substrate absolutely complements the structure of the enzyme, like puzzle pieces. Induced fit hypothesis. In this model, the enzyme, upon binding of substrate, changes shape. The matching between an enzyme's active site and the substrate isn't just like two puzzle pieces fitting together. Rather, the enzyme changes shape and binds to its substrate even more tightly. This fine-tuning of the enzyme to fit the substrate is called induced fit. Environmental effects on enzyme function. Active sites are very sensitive. They sense even the slightest change in the environment and respond accordingly. Some of the factors that affect the active site and consequently enzyme function include temperature. The suitable temperature for enzymes to function properly is 37 degrees Celsius. Increasing or decreasing the temperature above 37 degrees Celsius affects chemical bonds in the active site, making them less suited to bind substrates. Higher temperatures denature enzymes. pH. Amino acids present in the active site are acidic or basic. Fluctuation in pH can affect these amino acids, making it hard for substrates to bind. Extreme pH values can denature enzymes. Enzyme concentration. Increasing enzyme concentration will increase the rate of reaction, as more enzymes will be available to bind with the substrates. However, after a certain concentration, any increase will have no effect on the rate of reaction. Substrate concentration. Increasing substrate concentration increases the rate of reaction. This is because more substrate molecules will be colliding with enzyme molecules, so more product will be formed. But again, this effect is valid up to a certain concentration. Inhibition of enzyme activity. Some evil substances called inhibitors reduce or even stop the activity of enzymes in biochemical reactions. They either block or distort the active site, thus inhibiting the reaction. Based on this, there are two types of inhibitors given below. Competitive inhibitors. occupy the active site and prevent a substrate molecule from binding to the enzyme. Non-competitive inhibitors attach to parts of the enzyme other than the active site to distort the shape of an enzyme.
Yes, Manuti, are you ready with your presentation?
Yes, team.
Okay. Okay, then I will let you share the screen. So today, then I will discuss about enzymes on the next day because I think it is very heavy for you today. Then, Hari, then you can share your screen. Ina, I will let you share the screen.
Can you see the screen?
Yes, now we can see.
Good morning, everyone. Today I hope to present some facts about cyclones to you. In the presentation, I will define what a cyclone is and what are natural disasters, the factors causing cyclones and the three types of cyclones, and also reflect on how humans can deal with these disastrous cyclonic disturbances and help you with the quiz. What are natural disasters? Natural disasters are useless brought on human, animals, and property by natural processes without the mediation of man. What is depression and how does it lend the path for a cyclone to occur? If the low pressure area, which is created when the pressure of a certain area on the earth drops below the pressure around the plates, develops further, it becomes the depression. Then, if the depression develops further, there is a big potential to cause a cyclone. What factors cause cyclones? Existence of a large oceanic area and its temperature being about 27 Celsius to a depth of 60 meters, occurrence of convection current in the atmosphere, minimal tendency of the horizontally blowing wind to sweep vertically, location of the point at which the depression is closer to the equator, increasing humidity of the atmosphere beginning from the surface of the ocean to higher atmospheric levels. The three types of cyclones are tropical cyclones, which occur in the North and North Asia, typhoons, which occur in the North Pacific Ocean, and hurricanes, which occur in the North Atlantic Ocean. Structure of a cyclone. In addition to the circulation, air rises up in the central part of the whirl of the cyclone. This upward movement of air gives rise to a cylindrical cloud whirl. The central part of the eye whirlwind is called as the eye. It would have spread within a region of 30 to 60 kilometers from the center of the whirlwind. This eye, a region with little wind and free of rain and clouds. In satellite photographs, this appears as a black circle. The cylindrically arranged cluster of clouds around the eye is known as the eye wall. In this region, heavy rains and fast-blowing winds occur. Due to the eye wall, a few spotted bands of cloud can be seen. In these regions, too, heavy and speedy winds occur. More facts about cyclones. Cyclone is the main mechanism that distributes the huge solar energy received by the regions close to the equator of the globe. Cyclones originating time to time in Indian, Pacific and Atlantic Oceans provide the factors essential for the life of plants and animals on the earth. Though in nature, cyclone is a favorable phenomenon like this, today more attention is focused on the disasters brought by it. According to the further references, November and December are the months in which cyclones occur mostly. The most severe cyclone that made landfall in Sri Lanka in the past 12 months was the Bengal cyclone. How can we get rid of the damages caused by cyclones? Check whether the house is secured. Remove dead branches or dying trees close to the house. Keep some wooden boards ready for emergency and keep hurricane lanterns ready for function. Demolish condemned buildings. Keep the transistors ready with extra batteries. Be ready with the disaster kit and inform about victims as soon as possible to the relevant departments. And it's time for the quiz. What are the 3 types of cyclones?
Yes. What are the 3 types of cyclones, Janudi? Yes, Janudi. Oh, no response. Then we'll ask from Akshaya.
Tropical cyclone, humidity cyclone,
Right.
typhoon cyclone.
Okay. Then what next?
What are the 2 main months which cyclones occur mostly?
Yes, we'll ask from Shenali.
November and December.
Yes, Kian.
November and December.
November and December, right, Thesis.
What is the most severe cyclone that occurred in Sri Lanka in the last 12 months?
Bengal cyclone.
Bengal cyclone, right. Okay. So the newcomers may think what these presentations are. So we just concentrated on presentations from 6 to 9. But in grade 10 also we can continue. From 6 to 9 I gave the chance for 2 students. Now in grade 10, since we have a lot of things to discuss and we are taking about 5 minutes for the presentation, therefore we will take only 1 per week. He or she has to do it definitely. Mama Tama, I haven't listed out the names among list එකක් හදලා ඔයගොල්ලන්ට පිළිවෙලින් දෙන්නේ. පිළිවෙලින් කියන්නේ in order not to skip anyone. Therefore, I will make that and share with you from the coming week onwards. Right? We'll give the grade 10 topics also. Hari. So, then some of you, I think some of you are not joining with the paper class, but remember in grade 10, it is something essential, right? We are practicing to write answers for the O-level question paper here. තාම අපිට O-level questions වලට යන්න බෑ. අදට මම tute එකක්වත් share කරලා නැහැ because we are at the beginning of the first unit. So today also we have to discuss certain things which are important for you. I think Anissa, it is I can't say that it is compulsory. Some of you may have some other things, but remember it is something necessary because though you have the knowledge, you should know how to write the answers. ඒක ඉගෙනගෙන තියෙන්න ඕනේ කොහොමද උත්තරයක් ලියාගන්නේ කියන එක අනිවාර්යයෙන්ම ඉගෙන ගන්න ඕනේ. නැත්නම් කොච්චර දැනුම තිබුණත් ඒක කරන්න අමාරුයි. ස්කෝර් කරන්න හරියට දැනගෙන ඉන්න ඕනේ. ඒ නිසා try to join the paper class at 12:30. දැන් මෙතන break එකක් දෙනවා. පොඩ්ඩක් relax වෙලා එක දිගටම නෙවෙයිනේ මහන්සි නැහැනේ. ඒ නිසා join වෙන්න ගොඩක් දේවල් අපි එතෙන්දි කතා කරනවා. Harida. Okay then, since we have discussed so many things about the importance of carbohydrates, tests to identify carbohydrates, about proteins, test to identify proteins and the importance of proteins in the human body. We'll stop the theory part for today now, right? So we will meet again at 12:30, right? Okay then, see you and stay safe.
Thank you and God bless you.
Okay, bye.
Thank you.
Thank you.